Thank you very much. Our title of presentation is Growth and Yield of Sweet Corn as affected by incubation time of preparation for TITHONIA enriched liquid organic fertilizer. I'm Nani Setiawati from the Department of Crop Production, University of Bengkulu, Indonesia. This is our research team, Atika majoring in crop science, I myself, Nani Setiawati, majoring in wheat science, Fahru Rosi majoring in plant physiology, and Zainal Muktamar majoring in soil science. A serious problem in agriculture practice is the dependence of farmers on synthetic fertilizers, which are expensive and not environmentally friendly. An alternative to solve the environmental damage, especially the declining of soil fertility, is to use organic fertilizer. Organic fertilizer can improve soil quality because, in addition to increasing soil pH, it can also reduce exchangeable aluminum in the soil. Two types of organic fertilizer are solid organic fertilizer and liquid organic fertilizer. Liquid organic fertilizer contain essential nutrients for plant growth and easily dissolve in the soil. Source of organic material for LOF include plant residue, animal waste, and human waste, as well as utilizing local microorganisms such as vegetable and animal liquid waste. Unlike synthetic fertilizer, LOF contain easily available plant nutrients and generally does not harm our soil crop. On the other hand, solid organic fertilizer release plant nutrients slowly. Therefore, they are often not available when required. TITHONIA wheat is commonly used as a source of LOF. TITHONIA is a broadleaf wheat, grow fast, and contain high N. TITHONIA is easily decomposed in soil and provide high availability of N, P, and K for plant. A factor determining the quality of LOF is the duration fermentation time. In the fermentation process, organic material decompose and release nutrients for plant. The duration of fermentation affects the nutrient content of LOF. During the fermentation process, solid organic material decompose. The nutrients then are available for plant. The rate of decomposition is highly dependent on the microorganism activity. Microbial growth and reproduction depend on the duration time. Our objective of our study is to determine the growth and yield of sweet corn as affected by LOF prepared with different duration of incubation. The research was conducted from August to December 2019 at the Experimental Station Department of Crop Production, Faculty of Agriculture, Bengkulu University, Indonesia. The design of this experiment was a completely randomized design with one factor and replicated 5 times. The factor was time of duration of incubation consisted of 4, 5, 6, and 7 weeks, and control treatment without LOF application. This is the experimental process of our work in the field, starting with soil preparation, and then following by liquid organic fertilizer preparation, growing medium preparation, and then planting the corn, followed by LOF application in the corn. And then we did irrigation and pest control. You also do harvest and observe variable. Data were analyzed using ANOVA with F-test at the 5% level. This is our result of our study. Nutrient content of LOF at various duration of fermentation, and the analysis showed that there were differences in the nutrient content of each duration fermentation. LOF incubated for 4 and 5 weeks resulted in the higher nutrient content of N, P, and K than those of 6 and 7 weeks. The result may be associated with more intensive decomposition at 4, 5 weeks, where the decomposing microbial cell continue to grow. The decomposition of organic matter for LOF was accelerated at 5 weeks duration of fermentation, which was indicated by the increasing content of N, P, and K. At 5 weeks of fermentation, the content of N, P, and K was higher than that of 4 weeks of fermentation. The degree of acidity or pH is another factor influencing the activity of organism during the decomposition time. The lowest pH of LOF in this experiment has resulted from 4 weeks of decomposition. The pH increased as the fermentation time increased. The CN ratio is the ratio between carbon C and nitrogen N as indicator of the fermentation process. The result of this study showed that 4 weeks of fermentation resulted in a CN ratio of 9.05, which was lower than the other treatment. The all treatment had CN ratio below 14. Another factor affect the decrease in N content at 6 weeks of fermentation was the possibility of the evaporation of N. The stirring during the preparation of LOF may cause the loss of N through the evaporation. The effect of duration of fermentation on the growth of sweet corn resulted that LOF duration of fermentation had a significant effect on leaf greenness at 3 and 6 weeks after planting, and also for the plant dry weight. The growth of sweet corn was lower than the potential of the sweet corn variety Bonanza, which has plant height of 220 up to 250 centimeter and stem diameter of 20 to 30 millimeter. Those vermicompost applied as a base fertilizer has not met the nutrient requirement for sweet corn growth and development. The addition of LOF also did not increase the growth of sweet corn. Although the nutrient content of LOF was sufficient for plant growth and development, however, sweet corn plants have not fully utilized those nutrients. In this study, environmental factor might affect the nutrient availability for plant. High temperature during the study and very low monthly rainfall also influenced the plant growth. The LOF application at different duration of fermentation had no significant effect on the yield of sweet corn. The non-significant yield of sweet corn was related to the N, P, and K content of LOF might not be sufficient for the growth of the corn, also the development of the plant. Besides LOF treatment, environment plays significant effect on plant growth and yield. In this study, the abiotic factors such as high soil acidity, high air temperature, and low rainfall significantly affect crop yield. Acid soil affect the availability of nutrients, and the LOF application during the dry season with low rainfall and high air temperature might be ineffective due to the evaporation of LOF. This figure shows that LOF prepared for 4 and 5 weeks of fermentation tended to have higher average scope length than the other treatment. This result indicated that the LOF nutrient content prepared for 4 and 5 weeks of fermentation contributed to the scope length. The result of our study, then we conclude, firstly, that the LOF prepared for 4 and 5 weeks of fermentation had significant higher content of NP and K than those of 6 and 7 weeks. Secondly, the LOF increased leaf greenness and plant dry weight of sweet corn, even though had no significant effect on other variables observed in this study. That's about our study. Thank you very much for your kind of attention. Again, thank you. Assalamualaikum warahmatullahi wabarakatuh.